Okay, so welcome to this next video in which we are discussing intestinal stem cells. Okay, right. So, we have seen that the epithelium of the small intestine consists of these four terminally differentiated cell types. We have the enterocytes, which make up most of the epithelial cells of the small intestine, and these are the cells which are responsible for absorbing nutrients from the lumen of the small intestine into blood vessels that are within the lamina propria. Okay, then we also have goblet cells which are responsible for secreting mucus, which is going to cover the surface of the small intestine and lubricate it. Okay, then we've also got the enteroendocrine cells which secrete peptide hormones, but also other molecules such as serotonin. And then we've got the Paneth cells, which are the odd ones out, which are located right at the base of the crypts of Lieberkuhn, which are responsible for secreting antibacterial proteins, which keep the intestinal surface free of infection. Okay, right. So, all of these terminally differentiated cell types are not capable of dividing. They cannot divide. Okay, so that leaves us with a problem if we're going to need a continual supply of new intestinal epithelial cells, because they are continuously going through this conveyor belt procedure, okay, where they move up the villi, and then once they get to the tip they commit apoptosis and then pass into the lumen. Okay, so the intestinal stem cells are the solution to this. Now, they are not terminally differentiated. They can actually divide and produce cells of any one of these types. So there are 4 different types of cells that they can produce. Okay, so let's now talk about this in more detail. So, firstly, let's just have a little bit of terminology. These intestinal stem cells, because they can produce multiple different types of cells, okay, they are known as multipotent stem cells. So a multipotent stem cell is a stem cell that is capable of producing multiple different types of cells. Okay, contrast that to pluripotent stem cells. Now, a pluripotent stem cell is a stem cell that is capable of producing any type of cell within the human body. Okay, and there are around 200 different types of cells within the human body. So all cells of human body. Okay, these intestinal stem cells are not pluripotent stem cells. Finally, there's an even bigger term, which is totipotent stem cells. Okay, these intestinal stem cells are completely and utterly not totipotent stem cells. Totipotent stem cells are capable of producing all cells of the human body, but more than that, they can also produce all the cells of the placenta as well. So these are the cells which can actually become an embryo that is viable, because even though the pluripotent stem cells are the ones that you need to produce the embryo itself, an embryo isn't going to survive unless it can produce the extra embryonic tissues as well. Okay, so you need totipotent stem cells there. So these are all the extra embryonic tissues as well. Okay, so the notable example there would be the placenta. Okay, right. So those are just 2 other terms that you might see and get confused with. But these intestinal stem cells are stem cells that are multipotent. Okay, right. Now, let me discuss how these multipotent intestinal stem cells can actually produce new terminally differentiated cells. Okay, so if we have our intestinal stem cell here, which is down at the base of the crypt of Lieberkuhn, okay, not quite at the base, remember the Paneth cells are right at the base, but they're pretty far down, okay, this stem cell can divide, okay, so what it can do is it can divide into 2 new cells. So it's going to go through the process of division, and then the 2 daughter cells then have a choice, okay. They can either become stem cells, they can remain stem cells, or they can become differentiated cells, okay? And it is not the case that the two daughter cells of the stem cell here it's not the case that one of them has to become a stem cell and one has to become terminally differentiated. Both of them have a choice. They can either choose to become a stem cell, so this stem cell here, this daughter cell here, it can choose to become a stem cell like its mother here, or it can choose to become a terminally differentiated cell, so it can differentiate. This other daughter cell down here has the exact same choice to make, and both of them could become stem cells, both of them could go on and differentiate, okay? The probability that it will do either of these things seems to be a half, okay? But it is fundamentally a stochastic process, it's random, okay? The daughter cells of this stem cell could both become stem cells, they could both differentiate. One could become a stem cell, one could differentiate. And roughly, overall, if you look at this on a large scale, roughly, most of the time, half of them will end up remaining stem cells and half of them will differentiate. And that's exactly what you want, because you don't want them always differentiate, because then you'll run out of stem cells. You need some of them, or roughly a half of them, to remain stem cells so that you don't deplete your population of stem cells. Okay, right. So each of the daughter cells then has this choice to make as to whether it wants to remain a stem cell or whether it wants to differentiate. And we will talk a lot more in a moment about what determines whether the daughter cell remains a stem cell or it goes on to differentiate. Okay, now the important thing to understand is that once this daughter cell has decided to become a stem cell, that stem cell can't then just say, oh, I want to differentiate now. If it wants to produce differentiated cells, it then has to go through the division process. Okay, so it has to divide in two, and those two daughter cells then have the choice of whether they want to go on to differentiate or become stem cells again. Okay, so a stem cell doesn't just differentiate into a terminally differentiated cells. It firstly divides to produce two cells, which then both have the choice of whether they want to become stem cells or whether they want to differentiate. Okay, right. So let's say we've got our intestinal stem cell down here, which has just divided and it's produced two cells. Let's say one of them is going to remain a stem cell, and that's going to remain here, exactly where the mother cell was, and the other is going to get pushed upwards. Okay, and we'll see that actually the fact that there is a limited space down here, and some of them are ending up getting pushed up the crypt of Lieberkuhn, that's actually what's going to determine whether you remain a stem cell or whether you go on to differentiate, but we'll come back to that in a moment. Okay, alternatively, some of the cells will end up being pushed down, and those will become Paneth cells. Okay, right. Now, let's just discuss this a little bit further, because let's assume that this cell has got pushed upwards. Okay, so it's got pushed up the crypt of Lieberkuhn. Okay, it's now going to differentiate into a terminally differentiated cell. Now this process of differentiating into a terminally differentiated cell, and for drama I'll just denote the terminally differentiated cell like so, just to make it look completely different from the stem cell here. And I'll also colour it in a different colour. Okay, so here is our terminally differentiated cell that can't divide anymore. Okay, and here is our our daughter cell, this is, rather than a stem cell. Okay, we'll have the stem cell then in green. Okay, right. So, this process of becoming a terminally differentiated cell is not an instant process. You don't just say, right, I'm going to become a terminally differentiated cell now, and you don't click your fingers and it's happened. It takes time, okay? So, there is a lot of time between starting this process of differentiation and actually getting to be a terminally differentiated cell. However, once you have made the decision that you are going to differentiate it, you are then committed to it. You can't turn around, okay? And you are therefore known as a committed precursor. So the cells that are currently in this process of moving from there to there are known as the committed precursors. Okay, now the committed precursors are actually still capable of dividing, so it's only the terminally differentiated cells that are not capable of dividing. Okay, the committed precursors actually are still capable of dividing. Okay, so what's going to actually happen is, as you're going through this differentiation process, so let's say here we have our committed precursor cell that's on its way, it's now going to divide, and it's going to produce it's going to produce more committed precursor cells. Okay, so here it has divided into two, and both of these cells are now identical. They're both on their way through this path to becoming this terminally differentiated cell. So both of them will end up becoming the same cell. Okay, now for this reason, these committed precursor cells are also known as transit amplifying cells. Okay, because whilst they are in the transit between being a daughter cell of a stem cell and being this terminally differentiated cell, they're going to amplify their numbers. Okay, so this is a mechanism of amplifying the number of terminally differentiated cells that you're actually going to be able to produce. Okay, so these are known as transit amplifying cells. So to summarize then, we start off with our stem cell. It then divides, okay? Both of the stem cells, both of the daughter cells of the stem cell then have a choice to make, okay? They can either remain stem cells, and potentially both of them could end up remaining stem cells, okay? Or they could go down the pathway of differentiating. And whilst they're in the process of differentiating, they are committed, okay, so they can't get out of it anymore, but they are still capable of dividing, and they do indeed divide and produce multiple of these same committed precursors which are on their way through the differentiation process. And for that reason, they're also known as transit amplifying cells. So in fact, you'll actually end up with more than one terminally differentiated cell here. Okay, because in the process of transition, these cells have been dividing, and therefore you end up with multiple of them. Okay, right. Now, we'll come back to what decides whether a daughter cell actually is going to remain a stem cell or whether it's going to differentiate, but I've alluded to the fact that it's completely determined by whether it's stays in the position where its mother cell was, okay, which is near the base of the crypt of the epithelium, or whether it gets pushed upwards. Okay, so it's all about the position of the cell that determines that. Right. So, let's just discuss where the transit amplifying cells are. So, the stem cells here, the intestinal stem cells, they will divide. They'll produce the two daughter cells. Let's say one of these remains where the mother cell was, and therefore remains a stem cell. Okay, the other gets pushed up. Okay, so these portions lining the edge of the crypt of the epithelium here, the upper portion of the crypt of the epithelium, these cells which I'm colouring in blue, these are going to be the committed precursor cells, or the transit amplifying cells. So this portion of the crypt of the epithelium, this is full of very rapidly dividing cells which are on their way to becoming fully terminally differentiated cells. And each of these cells could become either an enterocyte, okay, in purple here, okay, that's most likely, or it could become a goblet cell in green, okay, or it could become an enteroendocrine cell. Okay, so here in yellow is the enteroendocrine cell, and let's say here is a goblet cell in green. Okay, and then as they finish the process of differentiation, they'll move out onto the surface of the epithelium, so they're no longer in the crypt of the epithelium. They move out of the crypt of the epithelium, then they'll make their way up the villi. This takes about 3 to 5 days before they get to the top of the villus, and then they'll commit apoptosis, and then pass into the lumen of the small intestine, and then they'll be defecated out. Okay, right, so that's the process of renewal of the epithelium of the small intestine. Okay, what I just want to mention is the fact that we also generate new Paneth cells. Okay, now the Paneth cells have much longer lifetimes than the other epithelial cells. So remember, once you've come out of the crypt of Lieberkühn, let's say this cell down here, this has got 3 to 5 days to live basically before it will have made its way up to the tip of the villus and then has to commit apoptosis. Okay, so they don't have very long lives. Whereas the Paneth cells, they live for a couple of weeks, okay, so for several weeks. So they have a much slower rate of turnover. Okay, but again, they are produced by the intestinal stem cells. So what will happen is an intestinal stem cell will divide, produce 2 daughter cells. If one of these daughter cells ends up being pushed down into the base of the crypt of Lieberkühn, then it will go through the differentiation process, which will turn you into a Paneth cell, and then you'll secrete proteins which are antibacterial in nature. Okay, right. So, what we now want to turn our attention to is the great story. The story of how it is decided whether you as a daughter cell remain a stem cell or whether you are going to go down the pathway of differentiation and become a terminally differentiated cell. So it's completely determined by whether you get to stay in this little location here, which is suitable for the life of intestinal stem cells, or whether you get pushed out. So if you get pushed up or if you get pushed down, you go down the route of differentiation. If you remain in this special location here, you remain an intestinal stem cell. Okay, right, so this special location, which if you remain in, you remain an intestinal stem cell, this is known as the stem cell niche, okay, or the intestinal stem cell niche. Now, what is so special about this location that maintains these cells here as intestinal stem cells? Well, there are many things that are special about this stem cell niche, but we are going to talk about one of them, okay? And one of the things that is special about this location is that you are very close to the Paneth cells, which are located just beneath you, okay? And these Paneth cells are going to be secreting special signaling molecules which tell the cells to remain stem cells. And if you as a cell end up being pushed upwards away from this supply of signaling molecules from the Paneth cells, you will no longer remain a stem cell and you'll go down the route of terminal differentiation. Okay, but remember, in order for a stem cell to go down the route of terminal differentiation, it firstly has to divide, and then the daughter cells could end up being pushed out, and then those could go down the route of terminal differentiation. Okay, so let's discuss this signaling molecule that's going to be released. So the signaling molecule that is going to be released by these Paneth cells are Wnt molecules. Okay, so Wnt ligand molecules. And these are going to be activating pathways within the stem cells which maintain them as stem cells. Okay, they're specifically going to be activating the Wnt beta-catenin pathway, okay, within the intestinal stem cells, and it is this pathway which changes gene expression and maintains you as a stem cell. Okay, now, the Wnt beta-catenin pathway is also known as the canonical Wnt pathway. Okay, so what we now want to discuss is this canonical Wnt pathway and how it changes gene expression within the cells and maintains these cells as the intestinal stem cells, because this is why only certain cells remain intestinal stem cells. In fact, the population of intestinal stem cells at the base of a crypt of Lieberkühn is around 15, so generally around 15 intestinal stem cells fit into this stem cell niche. When a stem cell chooses to divide, one of the daughter cells usually ends up being pushed out, whilst the other gets to remain in the spot where the mother cell was. The one that remains in the spot where the mother cell was, that will be exposed to the Wnt molecules and therefore will remain a stem cell. The one that gets pushed out, that's going to then go down the route of terminal differentiation. Okay, so we now then want to discuss the Wnt beta-catenin pathway then. Okay, so we'll start off by discussing the ligands themselves, the Wnt ligands. Okay, and I shouldn't have just put Wnt ligand because there are multiple Wnt molecules. Okay, so the Wnt molecules are all proteins, so I'll draw them like so. Here is a Wnt protein. Now, there are multiple different genes in mammals for Wnt proteins. In fact, there are actually 19 different genes for Wnt proteins. Okay, so let's just talk about these different genes so that you have some understanding of the fact that there is this huge diversity within Wnt ligands. Now, they will all have different sequences of organic bases in the gene, and they will therefore produce proteins with different sequences of amino acids. However, these proteins are similar enough that they will activate the same receptors, and they will activate the same downstream signaling pathway. So, let's discuss the name of the 19 different genes. Now, it's reasonably sensible nomenclature. I mean, every single one is called Wnt, followed by a number. Okay, so at least they're not all named after the different scientists who discovered them. But it's not perfect. If it was perfect, it would just be Wnt1, Wnt2, Wnt3, Wnt4, all the way down to Wnt19. It's not quite like that. Okay, so, the Wnt proteins then. Well, firstly, the genes. So there is the Wnt gene number 1. There are then 2 Wnt2s. There is Wnt2, and then there is also Wnt2b. Okay, there are then also 2 Wnt3s. There is Wnt3, and then there is Wnt3a. Okay, we'll go up here now. There is then 1 Wnt4. Okay, there are 2 Wnt5s, there's Wnt5a and Wnt5b. Okay, there is 1 Wnt6. Okay, there are 2 Wnt7s. Oh, whoops, where's the T gone in that? 2 Wnt7s, there's Wnt7a and there is Wnt7b. Okay, after Wnt7b, there are then 2 Wnt8s, Wnt8a and Wnt8b. Okay, there are then 2 Wnt9s, Wnt9a and Wnt9b. Then we have 2 Wnt10s, there is Wnt10a and Wnt10b. And then there is 1 Wnt11, so we're close to 19 now, and then it jumps. We don't have a Wnt12, we don't have a Wnt13, we don't have a Wnt14, we don't have a Wnt16, but, sorry, we do have a Wnt16, but we don't have a Wnt15. Okay, so that takes us up now to the 19 different genes for Wnt ligands that you find within mammals. Okay, so there are many different genes for these types of proteins. Okay, so that's just to show you the diversity of Wnt proteins. Okay, as far as we're concerned, they are all going to do pretty much the same thing, so we're just going to refer to them as Wnt. So when I write a box with Wnt in it, what it will mean is one of these. Okay, I do not really care which, although we will see later on that there is some importance with regards to the different Wnt ligands. They do bind to slightly different domains on the LRP5/6 proteins. Okay, right, so we'll continue our discussion of the Wnt beta-catenin pathway to try and understand how it's actually going to affect gene expression within our intestinal stem cells later on in the next video.